Hello everyone, and welcome to our video on quantifying MMP activity with image J and normalizing samples to an MMP standard. The goal of this segment of our series is to show you how to take a stain zymogram like this one and quantify the bands of MMP activity such that you can graph it as shown here. In order to do this procedure, you're going to need some basic software. Microsoft Office is what I use, but similar programs to PowerPoint and Excel will also work. I also use a free program called ImageJ to quantify the MMP activity bands, and you can download that at this link. And you're also going to need the scanned images of your zymograms before and after you stain them, and they should be good quality images, like around 300 dots per inch. In the last video we showed you how to use the molecular weight protein ladder in order to identify different MMPs in your zymograms. Now the next step is for us to quantify the bands of MMP activity in image J. So when you first open up image J, this is how it's going to look. You're going to see just a simple toolbar, and what you're going to do is go to File and Open, and you're going to scroll to the image of your zymogram, and from there you're going to hit Open. Now your image should appear, and you can zoom in and out of the image by holding the Shift key and pushing either minus to make it smaller or plus to make it bigger. So once it's about the right size, the first thing you want to do is have the rectangular tool selected, and this should be the default tool selected. And what you'll do is you're going to draw a nice long rectangle along the lane from the top of the gel to the bottom. So after you've made your selection, the next thing you're going to want to do is click Analyze, Gels, and select First Lane. So now that we have the first lane selected, all you do is you click in the middle of that box, you move it over to the next lane, and then you repeat the process. Go Analyze, Gels, and now we're going to click Select the next lane. Notice that there's a shortcut called Command 2. If you're using a PC, it's probably Control 2. So what you'll want to do is remember that, because that makes your job a little bit easier. So I'm going to click Select Next Lane, and now I'm going to drag the box to the next lane and use the shortcut to select the next lane. So for me, again, that's Command 2. So I've selected the next lane, and now I'm going to do this again for the next lane. Command 2. And now I'm going to do it again for the next one. Now notice, if I was going to put the box higher than the other ones, and I put Command 2, it actually snaps down to the same level, so I don't have to worry about vertical location of these boxes. What you really want to focus on is that you have the box in the correct location, such that you're not spilling into one of the neighboring lanes, and also you don't want the box to be so narrow that you're not getting the full area of each band. So the box size that I selected is just right, such that I'm getting the full lane and I'm not getting any of our neighboring lanes. So I'm just going to continue selecting the next of these. Finally, when you're done, what you're going to do is go back to Analyze, Gels, and now you're going to click Plot Lanes. When you click Plot Lanes, this window will appear. So before going to the next step, let me first explain what this is showing us. This is a graph showing the intensity of the bands that we have selected along the length of the lane. Okay, so as we go from the top of the lane to the bottom of the lane, what the program is doing is it's measuring how many dark pixels we have in each little slice here, and then it's plotting it. So you can see that the line here is very high because we have a lot of dark pixels, okay? And as we go down the lane, they're all dark pixels across the cross section until we reach this band of MMP activity. And so that's all dark, dark, dark until we reach that band of MMP activity. And now at this region, we don't have a lot of dark pixels. Now all the pixels have become light because of the MMP activity, which has cleaved away the gelatin, making us have less staining in our zymogram. So this lightness is corresponding with a drop in the intensity. And then once we reach the bottom of that band, which is right here, it becomes dark again. So you can see that we have dark pixels. We have a lot of dark pixels, so the line is high again. And then once we get to this other band, there's a drop in the intensity, and so forth. So you can see that if we want to get an idea of how big the area of activity is, all we have to do is somehow measure the area of this drop in intensity. And so what you do is you basically click and draw a straight line across the top of this peak. And from there we can measure the area of this shape, and that will correspond to the area of this band. Now this first lane is my MMP standard. And for my standards, I only measure the activity in the molecular weight range that corresponds to the MMPs that I'm interested in. So I'm only going to draw a line across this band. I'm just going to ignore this one for now. Now if we go down to one of the lanes that contain my samples as opposed to the MMP standard here, you can see that again we have a dark region corresponding to very little MMP activity. And then finally there's some light region, so there's the drop in intensity that we would expect to see. And again there's another little band here, which is this little drop in intensity right here. And then finally we're going to arrive at the band that we're interested in, which is active MMP2. So I'm going to draw a line across from here to here. And this is the area of my MMP2 which is active in this lane. Now I'm going to go to the third lane here, right here. So same pattern, and you can tell right below this band which we've selected up here is another smaller band, but it's at the same position along the length of the lane. So if you remember, from left here is the top of the lane and the right is the bottom of the lane. So as we go down the lane, we're going to find this band which is at the same location. So I'm just going to draw a line across and then continue downward to the next lane, draw another line across, and do that for the rest of the lanes here. And finally there's a very tiny one. This is, let's see, this is 1, 2, 3. So it's the third lane from the last one. And you can see that the third to the last one is right here. You can see that this is a very faint band, which makes sense. Here's the 10th lane. And then finally in the 11th lane, we don't even measure any MMP activity in that sample. So this is going to be a 0, all right? So now what's the next step now that we've closed off all these bands that we're interested in? So the next step is to go back to our toolbar here, and we're going to select the wand tool. And the wand tool is going to enable us to determine the area of each of these shapes that we've now enclosed. So now we have a way of measuring the area. So now we're just going to simply click in that first enclosure that we drew. And when I click, you can see that image J has measured the area in pixels of that shape, and it happens to be 12,879.811 pixels. So that first measurement was my MMP standard. Now my first sample, I'm going to click, and you can see the measurement has already been recorded. It's 4,800 pixels. You can do that here, here, going all the way down. Now, I've arrived to the last lane where I told you we couldn't really measure any MMP activity. As a reminder for myself, I usually just click in this open space, and when you get an absurdly large number of pixels, that's my cue to myself that this is a 0 measurement. And that's helpful if one of these middle lanes had 0 MMP activity. If you don't want to lose track of which lane had 0, just click on any empty space, And you'll remember that that absurdly large measurement is supposed to be just a 0. So you can use that as a way of keeping track, sort of as a placeholder for the future. So now what we've done is we've measured the areas which correspond to the area of MMP activity on our zymogram. So all we're going to do is copy this data, and we're going to open up Excel. So now that we've copied our data from image J, I'm simply going to paste it into Excel. Now I've already written here the lane numbers, and this column contains the measurements that we made of MMP activity in whatever lane and whatever band of activity that we're interested in. And I just made a note here to myself that the first lane is our MMP2 standard, which I've loaded into the gel for the sake of normalizing my data to it. I'm going to explain why I use MMP2 standard in the next couple minutes. And again, these are all of my samples. Now remember, this really large number is actually supposed to be a 0. And what that really means is that the MMP activity was so little that it was below the sensitivity of our assay. Now what we can do is we can straight away graph this data if we like, but there's one more thing that I'm going to do. The last thing I like to do is normalize my sample bands to an MMP standard. And the reason that I like to do this is because I've noticed over time that no two zymograms are the same. So in order to reduce gel-to-gel variation, I use an MMP standard. Here's an example of two zymograms which were used to measure MMP activity in very similar samples. And what I'd like you to notice is that there's a lot stronger bands of MMP activity in this zymogram than there are in this one. And you notice that for the active MMP2 bands, the trend is also the same, that these bands just happen to be a lot brighter than these ones, even though the samples that were put into each of these zymograms were quite similar. Now, when I set this zymogram up, I added 10 nanograms of a purified MMP2 standard. And on our website, we have instructions on how you can prepare a standard like this. And it's very useful because you notice that in this zymogram, which has stronger bands of MMP activity, the standard also seems to have stronger activity. So if we just take the MMP activity that we measure in each of our samples and divide it by the MMP activity in our standard, all of our samples are going to be relative to 10 nanograms of MMP2. That way, if we're trying to compare samples in this zymogram with samples from that zymogram, we're not going to have variations in our measurements just due to differences in the zymograms themselves. Everything is going to be relative to a standard. So it's really a very simple calculation. All we're going to do is we're going to take the MMP activity of each sample and divide it by the activity of our standard. If we're going to do that for our first sample here, all we're going to do is hit the equal sign, click on the activity of that sample, and then the divide by sign, and then click on the activity of our standard and hit enter. So there's the calculation right there. We can repeat that process for all of these cells, but that would be a little bit repetitive. If we want to take a shortcut, we can make use of this little dragging feature in Excel. But before we do that, we first have to freeze the cell that corresponds to our standard. So that way when we drag the function down, then we're not going to also drag down the cells containing our standard. We want that standard to be the same for all of our samples. So it's very simple. Just put the $sign in front of the number 4 corresponding to the row number that contains our standard. Once you put that $sign, all you have to do is select the cell again and drag this little box in the bottom right hand corner down. And what you'll find is that, let's say we look at the next sample, the formula is exactly how we want it to be. We're dividing B6, which is the intensity of our sample, by B4, which is the intensity of our standard. And if you double check any of these other samples, it's going to be the same thing. So now we've finally accomplished what we wanted to, which is we've identified what MMPs might be in each of these bands, and then we've quantified the activity in each of them, and we've normalized the activity to a control, a standard, which we've added to each of these zymograms, in order to account for gel-to-gel variations. So with that, I hope you stayed tuned, because in the next video, we are going to show you how you can take that data which you've just quantified and turn that into publication quality graphs. Thank you so much for watching this video. If it benefited you, please remember to click the like button. And if you're interested in seeing more of our videos, be sure to subscribe to get updated when new videos are released. Thanks for watching.